So let's start by calculating our residuals. Okay, and let me just show how we can compute these residuals there. Now remember, this is a multiple regression, so this can fit any practical work. Okay, so I'm going to plot the gene multi. Okay, plotting the entire gene multi. Okay, let me show you how it shows on the right side here. So this is a plot of the gene multi, the whole of the plot. And this is everything. This is the residuals. All the residuals versus the fitted. Okay. And you can see the plot, the actual plot in blue. The residual versus the fitted, that's what you see down here. The gap between the residuals and the fitted. One is a dot, and one is a red line rising, falling around the dot. It will help you, this plot can actually help you to know observations that can cause problems. So on the top right, you see that observation number 24 is causing a possible problem. We wouldn't know until we check the influences. And all the independent variables are down here. They are the part of the fitted values. Okay, so that is a quick plot that you can do. Then the next one is that you can use the par-inflow command to commandeer that every other plot that I'm about to do, that plot, I want you to be able to put it into 4 parts. So I don't want you to give me 1, 1, 1, but I want you to put all of them into all the 4 charts into one panel. Okay. The plots we are interested in will be the plots at the top left. And first, let me generate the plot there. Okay. So I'm just going to do par-inflow, and then I now run the plot. Once I click run on the right here, you will see the plots. It's beautiful. So the first plot is the residuals versus the fitted plot, something I showed you earlier. Okay. The second plot on the top right is the normal quantile probability plot. Now, the most important thing of this is that the dots must sit on the line on average. If generally they are around the line, it means that there may be no residual problems. Okay. Problems related to variance of the error term. Now at the top, you see again observation number 24 appearing. It appears to appear in every graph, including the scale location and then the residual versus the leverage. Okay. Now these ones are used to check for homoscedasticity, which we will later come to analyze. But this is a final aspect of the multiple regression that we are looking at. Okay, before we go into the details of the assumptions, valuations. But this is just to give you, okay, in the top left one here and the bottom left one here tells you those guys that can be influences. Again, you could see that the fitted values and the residual values seems to be moving in the same kind of direction or closer to each other, as you can see that. So R has been able to do all of that. But next time, what we are going to do is that we are going to just build beautiful results. Beautiful results, which is very nice because it helps you to be able to understand a lot of other things. But other than that, this is how you finish the extra things to do when it comes to multiple regression.